Today I'm going to explain the homeostasis and transport of auxin. Auxin signaling pathway. Auxin's effects are dependent upon its accumulation in the plant cell. The combined effects of transport, biosynthesis, conjugation, and degradation ensures that this occurs. The auxin response pathway is maintained by feedback regulation and also by environmental factors and other hormones. The biosynthesis and homeostasis of auxin. Auxin can be made from IAA. IAA is synthesized in two ways. The main pathway involves tryptophan and many other pathways. The first of these pathways is the IPA pathway. This pathway converts tryptophan to IAA via indole-3 pyruvic acid. The second pathway is the IAM pathway. This pathway converts tryptophan to IAA via indole-3 acetamide. And the final pathway is the indole-3 acetoxamide pathway, or the IAOX. And this pathway converts tryptophan to IAA by using IAOX. The second way to make IAA is by synthesizing IAA from indole. This pathway is tryptophan independent, and the steps of this pathway are not well understood. As I mentioned before, auxin synthesis is regulated by other hormones, such as jasmonates and ethylene, and by environmental factors, such as temperature. Another important aspect of auxin homeostasis involves conjugation and degradation. Conjugation and degradation of auxin also regulates its homeostasis. The GH3 gene encodes for auxin conjugating enzymes. The overexpression of these enzymes reduces auxin levels in plants and causes a dwarf phenotype to be produced. The reduction of auxin implies that conjugation is a part of a negative feedback system. So to summarize the auxin homeostasis, we know that homeostasis of auxin is tightly controlled by its synthesis, catabolism, conjugation, and transport, and is also influenced by environmental factors and other hormones. Polar auxin transport. Auxin is transported from one plant organ to another by polar transport. Polar transport simply means that auxin is transported cell to cell in a polar direction. Transport of auxin can also occur via auxin transport proteins. Movement of auxin is from top to base, also known as basipedal, followed by movement from base to the tip of the root, known as acropedal. At the root tip, auxin moves back up the root again, but only for short distances. Graphotropism in plants is due to auxin. Plants are graphotrophic. This simply means that plants respond to changes in their orientation. Shoot, it is negatively graphotrophic. This means that the shoots grow away from the center of gravity or up toward the sun. On the other hand, roots are positively graphotrophic and grow towards the center of gravity, which is really down into the soil. Differential growth due to auxin levels. Auxin accumulation promotes elongation. Auxin accumulates on the lower side of a plant coleoptile, which causes bending upward on the upper side. Another important aspect of auxin accumulation is that increased accumulation leads to greater growth. Increased accumulation of auxin causes greater growth in the coleoptiles. As indicated in the diagram, the lower side of the coleoptile has greater growth because it has greater accumulation of auxin compared to the upper side. So, summarize. Transport of auxin is polar from basipedal to agropedal, but auxin can also move via transport proteins. Movement of auxin is essential in pattern formation, organogenesis, and directional growth responses.